Cancer is not simple. It can come from a variety of different factors, but the biggest points of control is growth signals and, hey, hit the brake on the cell cycle signal. Proto-oncogenes are for growth, and oncogenes are cancerous out-of-control growth, and tumor suppressors are to put a pause on it and hopefully fix it or kill it before it becomes a problem. The MAPK signaling sequence is a complicated series of signaling where oncogenes or defective tumor suppressors can arise to result in cancer, as it is the signal relay between the outside of the cell and the nucleus to get mitosis going. The cell has a lot of information going on from the outside all the time telling it to do stuff. So one of these signaling molecules floating around the outside cellular space finds its way onto the ectodomain or outside domain of the receptor tyrosine kinase, or RTK, causing it to dimerize and bring the kinase domains close together to cross-phosphorylate the RTK. Proteins inside the cells have SH2 or PTB domain to recognize and dock onto the phosphorylated RTK. Proteins with SH2 to bind to the RTK includes PI3 kinase or CSER. But the one we're looking at is GRAB2, which gets onto the phospho-RTK and recruits SOS. SOS stands for Son of Sevenless, not Save Our Souls, which to me sounds like an awesome name for a pirate crew, so I'm drawing it to be a pirate ship. So, SH2 binds to RTK, and the SH3 domain binds to SOS, and SH3 has a proline binding domain, and the SOS is full of proline, so we just stick that on there. When SOS is bound to GRAB2, it goes to RAS, and SOS is a GEF, or guanine exchange factor for RAS, trading GTP and GDP to activate RAS. GEF is also involved in punching the tRNA through the ribosome for translation, so I think of SOS as punching the GDP out of RAS and leaving a GTP on it. RAS with a GDP is inactive, while RAS with a GTP is active. RAS shouldn't be active forever, so there's a RAS gap, NF1, with a GTPase using hydrolysis to convert GTP into GDP to deactivate RAS. RAS could convert GTP to GDP on its own, but it takes too long for effective signaling, so it uses RASGAP. Anyways, active RAS goes to activate RAF, which activates MEK via phosphorylation, and MEK activates ERK, also with phosphorylation, so it's RAF, MEK, ERK. This part specifically is the MAPK pathway. ERK then goes on to activate transcription factors by phosphorylating Phos, MYC, and Jun in the nucleus to make them start transcribing mRNA to make the needed proteins. However, the MAPK cascade can't be left on forever, so to turn it off, ERK is also able to phosphorylate SOS and RAF, which deactivates them both. ERK is also results in DUSP being transcribed, and DUSP dephosphorylates and resets the RTK, and then there's the previously mentioned RAS gap turning off RAS. All of this is able to shut down the MAPK cascade when the job is done. But why is this important? Well, things going wrong in a couple of these steps can result in uncontrolled cell proliferation, the kind of errant replication that makes cancers malignant. Really bad. So keep complicated MAPK in mind as one of the reasons why cancer is complicated and hard to find the cure to. Anyways, that's the MAPK pathway. Thank you for listening.